Thank you and good morning. So I'm Stephanie Chertsky, co-founder of Papillon Therapeutics. So our company focuses on the multisystemic gene therapy for life-threatening condition. And the technique that we are using is called autologous hematopoietic stem cell transplantation of ex vivo gene-modified cells. So what we do is we collect the CD34 hematopoietic stem cells from the blood of a patient, and we ex vivo gene-modify using lentivirus vector or CRISPR-Cas9 gene editing. Once the cells are gene-modified, they're cryopreserved, characterized, and if they meet the release criteria, they are reinfused into the patient after a short-term myeloablation. So the stem cells reconstitute the bone marrow, and they can give rise to all the blood cells. So this approach is well known to treat immunodeficiency or all kind of blood disease. But there is a blood type that is very interesting into these blood hematopoietic cells is the monocytes. The monocytes have the ability to differentiate and engraft long-term into the tissue, and they differentiate in macrophages or macroglia into the CNS. And so in Papillon, using this approach, we have several programs. So the first program we worked on was called cystinosis. And for this program, we went from the preclinical proof of concept all the way to phase 1 clinical trial. And we have now fully enrolled the phase 1/2 trial, and this program has been recently acquired by Novartis. So the second program is on Friedreich ataxia, and for that we are at the stage of IND-enabling studies. And for Danon, we are just entering actually the IND-enabling studies. And we have a last program that is at the proof of concept preclinical stage, which is for Alzheimer. So I will review all this program quickly. So for cystinosis, the disease is due to mutation in the CTNS gene that is ubiquitously expressed, and it encodes a transmembrane lysosomal transporter of cysteine. And in patients, this transporter is absent, and so the cysteine accumulates into the lysosome. And because it's expressed in every single tissue, we have a progressive degeneration of all the organs in these patients. And so what we've used in this, using ex vivo hematopoietic stem cells that was very unexpected, is that even in the context of a transmembrane protein and in a disease really impacting all the organ, we were able to obtain a very beneficial effect and preservation and stopping the disease actually in the mice. And we see also positive results in the patient now, with all the patient, most of the patients being off their cysteamine drugs. And even after more than 3 years post-stem cell infusion. And we have reduced cysteine accumulation in the blood, but also in the organs. And we have a clinical improvement for the CNS and muscle strength, etc. And we have also a very good safety and tolerability profile for this technique. So this is the program that now, as I said, is acquired by Novartis. So with what we've learned with cystinosis and the mechanism of action, we realized that we can go much further with this ex vivo gene therapy process. And we decided to go into the realm of mitochondrial disease. And as a proof of concept, we decided to start with Friedreich ataxia, which is a neurodegenerative disorder due to a mutation, expansion mutation in the intron 1 of the frataxin gene, resulting in the decrease of expression of this protein. Because frataxin is a mitochondrial protein, this leads to cell death, and as I said, neurodegeneration and muscle weakness, so the patient lose their ambulatory function, and eventually they die from cardiomyopathy. So the drug product that we want to bring to clinic is actually the gene modification of the hematopoietic stem cells using CRISPR-Cas9 to remove the GA expansion in the intron 1 of frataxin. And this is very advantageous for many reasons. First of all, it's all the patients actually carry this mutation. So we will have the same reagent and the same manufacturing for all the patients. And the GA repeat is in the intron 1, so if there is a mutation at the site of the cut, it's not an issue. And the gene stays under the regulation of its own promoter, which is a key factor for this disease because overexpressing frataxin is toxic. So when we did the proof of concept using hematopoietic stem cells that you can see here in green, I don't know if you can realize how much the hematopoietic stem cells, so here the hematopoietic stem cell differentiated in macroglia into the CNS, and we can find them very abundantly in the brain, spinal cord, and also dorsal root ganglia. So the cells really migrate where they have to migrate in the damaged tissue. But they also can go to the heart and muscle. So in one-time treatment, one-time bone marrow transplant, the patient will receive a constant provision of a healthy protein for the life of a patient. And that's a very key point of ex vivo gene therapy. And so for showing disease-modifying activity, we used the mouse model of Friedreich ataxia. And this mouse model has the advantage to be a knockout for the murine frataxin, and it carries the human transgene with a GA repeat. So we can use all the reagent that we are using in the human cells into the mice model. And in the mouse model, the hematopoietic stem cells are actually more difficult to gene edit than the CD34 human cells. So we can reach only between 10 to 20% of gene editing in the murine cells. But despite this fact, we see a good number of gene-edited cells in all the hematopoietic tissues, but also in all the non-hematopoietic tissues, all the tissues impacted by the disease. But most importantly, we see a significant and dramatic increase of the expression of the protein in all these tissues. And this has never been achieved by any other program. But most also importantly is that we have shown benefits. We have showed that the cardiac hypertrophy that we see in this patient into the non-treated mice or mice treated with the disease cells, they both showed cardiac hypertrophy. And this is rescued into the mice receiving the gene editing cells. As I said, the other major hallmark of the disease is the neurodegeneration, and we show that we can stop that in the spinal cord, but also in the cerebellum here, that you can see in the granular cell layer that there is a reduced number of neurons into the non-treated or mice treated with the disease cells, whereas the number of neurons is completely rescued in the mice receiving gene-editing hematopoietic stem cells. So as I said, for the Friedreich ataxia program, now we are in the IND-enabling studies, and we are hoping to move to a clinical trial in the next couple of years. So now the next program is the Danon disease. So the Danon disease is also a lysosomal disease due to a transmembrane protein like cystinosis, and the gene involved is LAMP2. So if in the patient, because LAMP2 is absent, there is a disruption of the autophagy. And this is very impactful in the heart. And actually, the patient develops a severe cardiomyopathy and all require heart transplant. But they also have skeletal myopathy and intellectual disability. So in this case, the drug product is the hematopoietic stem cell ex vivo modified with a lentivirus vector, and we are using the same lentivirus vector that we use in our cystinosis program, for which we have a good safety profile. And here again, in our preclinical studies, what we show is that all the mice showed a good rescue of expression of in the protein in the whole tissue impacted by the disease, like muscle, brain, but also in the heart. And what is very interesting is that we see the LAMP2 protein expressed in the macrophages that what we can find in the heart that are derived from the hematopoietic stem cells, but also the LAMP2 protein can be seen in the cardiomyocyte, showing a transfer of a LAMP2 from the macrophages to the cardiomyocyte. And this is what we also showed in Friedreich ataxia, that the protein is transferred to the neuron. And that's why the hematopoietic stem cells are very efficient into this disease. So after we showed that the cardiac function is improved into the mice receiving the gene-modified hematopoietic stem cells, as well as the cognitive function, and much more. So the last program I wanted to touch base is we have only very preliminary proof of concept, but this data can be found in a recent paper that we just published in Cell Reports. So in this case, we are using wild-type hematopoietic stem cells derived from GFP transgenic mice. And as control, we are using non-disease mice or mice receiving disease cells. And in this case, our mouse model of familial Alzheimer, which carry mutation in the APP and PS1, presenilin-1 genes. So what we have seen is that we can completely rescue the neurocognitive function in these mice. And you can see that in the two middle groups, you can see that the memory function is very reduced. So we have a non-disease mice, or the mice receiving the disease cells that have no benefits at all into the mice. Whereas that the mice receiving the wild-type hematopoietic stem cells, you can see a full rescue of the memory. And in the Y maze, we can see that they have also a full rescue of a sensation of fear, whereas the other mice with Alzheimer's have none. We have also shown that we can reduce the A-beta amyloid plaque into these mice. So you can see here a test mice in the lower panel and in a mock mice receiving the disease cells in the higher panel. So we can reduce that. But most importantly, and I think this is the key in this in Alzheimer's disease, and also a key feature in the pathogenesis of this disease, is that we completely prevent the neuroinflammation. Indeed, the mice, untreated mice or mice receiving mock disease cells, the macroglia are very activated, and the activation of macroglia leads to neurodegeneration. So by stopping this neurodegeneration, we believe that this is a key point of why we are preserving the mice from neurodegeneration. So I hope I'll show you the power of ex vivo gene therapy. We showed in a multi-organ degenerative disorder, even in the context of a transmembrane lysosomal protein, that ex vivo gene therapy could have a beneficial effect, and we've shown that in preclinical studies, but all the way also in our clinical studies, and this over 3 years post-transplantation. But we have also really good data in a mouse model for our next program, which is Friedreich ataxia, which is part of a mitochondrial disease, Danon disease, also lysosomal disease, but also in Alzheimer. So we hope that when we can really make a difference for this disease too. So on this, I just want to thank you for your attention.